Lasker Koshland Special Achievement Award in Medical Science for bold, imaginative, and diverse contributions to medical science and human rights. She discovered the BRCA1 gene locus that causes hereditary breast cancer and deployed DNA strategies that reunite missing persons or their remains with their families. Honors Mary Claire King.
For being such a trailblazer and for showing that you can be a passionate scientist and a humanist at the same time.
I would say that she's one of the leading human geneticists of her generation.
Mary Claire King goes to graduate school with the intention of studying statistics, but her career pathway diverts when she's introduced to genetics by Kurt Stern.
This is the most beautiful way of thinking. I just love this. It's mathematical in the sense that it requires being rigorous. And I asked Dr. Stern if it was possible to transfer to genetics, and he said, of course. And I did, and I've never looked back.
Working with Alan Wilson, King develops a research project for her PhD. Her challenge is to identify the differences in the proteins found in the blood of humans and chimpanzees.
Every time I would make a comparison, I would see either no difference or very subtle differences. And it was very, very rare that I would see a protein that was dramatically different in its charge or in its size between the human blood samples that I had and the chimpanzee blood samples that I had. And of course I thought it was me. I thought, I can't do these experiments, there's something wrong.
King tries multiple other methods available in the 1970s and still comes to the same conclusion, which she and Wilson publish in 1975. Contrary to mainstream beliefs at the time, their findings indicate that human and chimpanzee proteins are more than 99% identical in sequence.
Our hypothesis from this was that the differences between humans and chimpanzees that have led to these dramatic differences in morphology and behavior are not due to sequences of DNA or amino acids that encode proteins, but to individual changes that regulate the, as we said, tempo and mode of expression of genes.
The idea that breast cancer could be related to a specific gene was not widely accepted. There had been earlier studies that said that there was a genetic component.
However, King sees repeated observations from the time of the ancient Greeks to Paul Broca, noting clusters of breast cancer in families.
Very good.
She turns to genetics for the answers.
It took until 1988 to have a full test of the hypothesis statistically, and to be able to present a model that statistically, mathematically, was by far the best fitting model that postulated an inherited form of breast cancer due to a single mutation in a single gene in each family.
King sets forth to actually prove the hypothesis which she has elucidated from her mathematical model.
So in order to prove that this was true, I was going to have to actually have an experimental result. And the best way in the 1980s to prove that a gene existed was to figure out where it was, to map it to a chromosome. In other words, to give it a physical reality, an address, a physical location.
King is able to locate the gene to a particular stretch of chromosome 17, called 17q21.
We didn't have the sequence of the gene. There was no genome sequence yet. There was no reference sequence you could go to, but we knew chromosomes and we knew signposts.
King is thrilled when the head of the Committee for Nomenclature of Human Genetics asks her to name the gene.
And I said, well, I'd like to name it for Paul Broca. And she said, well, genes can only have 4 letters in their name, and Broca has 5. Could you consider a 4-letter name? And I said, well, how about BRCA1 for breast cancer 1? And she said, that's a lovely name.
Thanks to a scheme developed by King to screen for harmful versions of genes which predispose women to breast and ovarian cancer, doctors are now able to provide genetic testing for their patients.
It's progress. It's remarkable progress. If technology is is made available to young women, there should be no woman ever again die of breast cancer due to inherited mutations in BRCA1 or BRCA2 or any of the other sister genes.
So thousands of women are impacted by this research, and over generations, millions of women will be impacted.
A prolific problem solver dedicated to helping mankind with her skills as a geneticist, King doesn't hesitate when she's contacted by the American Association for the Advancement of Science to help the grandmothers of Argentina searching for their kidnapped children and grandchildren.
I thought that the assignment would be one or a few trips, that it would be a symbolic gesture, that we would show solidarity. It turned into a 30-year collaboration, the development of technology that was exactly what the grandmothers needed for the identification of their grandchildren.
To identify the grandchildren, King relies on the analysis of mitochondrial DNA, cellular components passed from mother to child that connect people to female ancestors. This technology is ultimately adopted by many in forensic science.
And the grandmothers have now identified and enabled the return of 108 children, who of course are now adults in their 30s.
She would go to Argentina to help these powerless grandmothers find justice. And that's really what's so special about Mary Claire and the work that she did in Argentina, and the work she continues to do to help human geneticists all over the world use the power of genetics for social change.
In the 1970s, when Mary Claire King begins her journey in genetics, many of today's technologies don't exist. Her unique breakthroughs are all the more valued for her critical thinking and the perseverance through decades.
Here was this absolutely brilliant woman scientist trying to set new paradigms for disease, blaze new trail. It must have been very hard for her, but she did it, and we celebrate that now.
She gets on that problem and she wants to solve that problem. She's accomplished wonderful things at each stage of her career. She's very determined woman.
A lot of it was good luck at many, many steps. Did I have a life plan? No. I was trying to make a living for my daughter and me, but I was lucky.